In this session, we will study about the functions of proximal convoluted tubule, then Henle's loop and DCTs and even the collecting ducts. And then we will also see about urine formation. Now, before I get into the topic, so in the last session, I was talking about the Bowman's capsule, and I told you that the filtration takes place through three layers. So one will be the endothelium of the capillaries, then the next layer will be the basement membrane, and the third one will be the epithelium of the right Bowman's capsule. Now, okay, so here the epithelial cells of that Bowman's capsule are what we call them the podocytes. Now, how these podocytes are present, and okay, here actually the podocytes will wrap around the capillary. So say suppose, okay, so this is the capillary, right, and this is the podocyte, and all these fingers are the projections, that is cytoplasmic projections of that podocyte. Okay, and this is the capillary. So now the podocyte will wrap around this, okay, the capillary in this way, and you can see that the cytoplasmic projections, in between this there is a gap, and you can see the capillary here, right? So this is how the podocyte will wrap around the capillaries. So all it will be wrapping in this way, and you will find, okay, very minute gaps between the cytoplasmic processes, and these are called the slit ports, or also called the filtration slits. So here I take the capillary, and here you will find, okay, so the nucleus of the podocyte, and you can see here, I'm just drawing, okay, and so this is that gap between the cytoplasmic processes. So cytoplasmic processes of the podocyte. So this is the podocyte cell. The podocyte is nothing but the epithelial cell of the Bowman's capsule. So you can see the how it is wrapping around the capillary blood vessel, and here this is the nucleus of the podocyte, and you can see the gaps between these cytoplasmic process. Of course, okay, it looks like a very big here, but they are very minute spaces, and these are what we call them as the filtration slits or slit ports through which the process of filtration takes place, right? So this is how the podocytes will be seen wrapping around the capillaries in the glomerular network that is located in the Bowman's capsule, right? So now that's about, okay, how these epithelial cells are present wrapping around the capillary walls. The next one we will see about the functions of these tubules, okay, the proximal convoluted tubule and Henle's loop and the DCT and even the collecting duct. So first, so we'll see about the proximal convoluted tubule. So in short, we call it the PCT. So here, this proximal convoluted tubule is lined by simple cuboidal epithelium, lined by simple cuboidal epithelium, and this simple cuboidal epithelium will be, okay, with the brush border, with brush border. Disorder is due to presence of microvilli, due to presence of microvilli. We know, we have seen what actually are these microvilli, which are also present in the small intestine. So if you see the cuboidal cells, so you will find the cuboidal cells in this way lining the PCT. So the PCT has a absorption function, so it reabsorbs a lot of material. So for that reason, uh, the cells lining the PCT will have the microvilli, which will help in increasing the area of absorption. So you'll find here, okay, the nucleus of the cells, and here the free surfaces will show this microvilli, and these microvilli will give the appearance of a brush. So that's what we say, it's a brush border. Okay, so these are all the microvilli, so that we call it the brush border. Okay, so this is the cuboidal epithelium, and you will find the basement membrane. Okay, so the proximal convoluted tubule is lined, okay, by the cuboidal epithelium that has the brush border, and this brush border actually helps in increasing the surface area for absorption. So now let's look at the functions of this PCT. So what happens in the proximal convoluted tubule? So there in PCT, 70 to 80% of electrolytes, so 70 to 80% of electrolytes and water is reabsorbed. So we know that the filtration takes place in the Bowman's capsule. From there the filtrate enters into the PCT, and in PCT, okay, these electrolytes and water will be reabsorbed. The electrolytes will be like potassium, bicarbonates, etc., are the one who will be reabsorbed back, okay, into the blood circulation. So this is one point. Then coming to the next point, almost all essential elements will also be reabsorbed. Okay, so almost all essential elements are also reabsorbed, and these essential elements will be like okay, potassium, then phosphorus, okay, magnesium, iodine, and etc., will be the essential elements, and almost all these essential elements will be reabsorbed in the PCT. Then right, then this PCT also will help in maintaining the pH and ionic balance of the body. So PCT helps in maintaining, okay, the ionic, okay, and also the pH of the body, and this it will do it by the process of selective secretion of hydrogen ions. So it will help in maintaining the ionic, okay, fluids, I mean sorry, ionic right substances, and also with that maintaining the ionic okay balance, it will help in maintaining the pH of the body. Okay, uh, and it will be doing it by a process called selective secretion. So it will be by selective secretion. So later we'll see what actually is this the selective secretion. And this selective secretion means it selectively releases certain substances like these hydrogen ions and the ammonia, okay, and potassium ions into the filtrate. So it will release these hydrogen ions, ammonia, potassium into the filtrate. So actually, right, which I will discuss it later. The process of this urine formation will have, okay, four steps like the filtration, reabsorption, okay, secretion and excretion are the four, okay, processes that take place during the formation of urine. And there, okay, so selectively some substances will be, okay, released back into the filtrate like these hydrogen ions, ammonia and potassium ions. So with that it will help this ionic balance, thereby maintaining the pH, okay, of the body. And at the same time releasing these substances, okay, at the same time it also absorbs, okay, the bicarbonates from the filtrate, bicarbonates from the filtrate. So that's what we'll see here. Okay, right. So when we see here, so selective secretion means releasing into the filtrate. So whatever inside this PCT is what we call it the filtrate. So into this it releases these hydrogen ions, ammonia, okay, and K plus ions, okay, etc. And from the filtrate it is going to absorb the bicarbonates, okay. This is how the PCT will help in the ionic and the pH balance. Now next one we'll see about the Henle's loop. The next one, Henle's loop. Okay, so how the Henle's loop functions. Here this is what we call a hairpin loop. So it's a hairpin loop. So we can see here, so the PCT becomes narrower, and this is how you will see this is the Henle's loop. So this Henle's loop has a descending, okay, loop of Henle, and this will be the ascending loop of Henle. So descending and ascending loop of Henle. So this is the descending and this is the ascending loop of Henle. Here, when you come to the juxtamedullary nephrons, in juxtamedullary nephrons, the ascending loop will have two segments. One is thin and another one is thick segments. So I'll just mention here the juxtamedullary nephrons. So in case of juxtamedullary nephrons, they have, okay, I mean especially talking about ascending limb of loop of Henle. Okay, so I'll just mention here ascending limb of loop of Henle. This is again, okay, right? So this is a descending loop of descending limb of loop of Henle, and this is ascending limb of loop of Henle. And coming to this juxtamedullary nephrons, the ascending limb of loop of Henle will have, so it has one thin segment, and also it has a thick segment. It has thick segment. So if you see the diagram on the page number 295 in your textbook, so he mentioned both the thin and thick segments for the nephron. It means it is the juxtamedullary nephrons. He has shown, okay, the juxtamedullary nephron. Whereas in the case of cortical nephrons, the cortical nephron will have only the thin segment. Okay, right. The cortical nephrons, they have only thin segment. Only in the case of juxtamedullary nephrons, you will find two parts for this ascending limb of loop of Henle. One will be the thin, this will be the lower, okay, thin, and a little bit upper, okay, thick segment. So thin and thick segments are seen only in juxtamedullary, whereas cortical has only the thin segment. So now let's see the functions, okay, of this Henle's loop. This Henle's loop is lined by, okay, squamous epithelium. Is lined by squamous epithelium. Okay. So now let's look at the functions. Right. So this segment will play an important role in maintaining high osmolarity. So it helps in maintaining high osmolarity. So this high osmolarity will be in the medullary interstitial fluid. In the medullary interstitial fluid. Suppose you are seeing so this is the Henle's loop. Okay, so this is the descending, okay, loop of Henle, and this is ascending, okay, limb of loop of Henle. And whatever this, you will see the fluids here. So the space between the ascending limb and the descending limb, okay, so all it is filled with the fluid. And we know that some I mean the Henle's loop is present in the right medullary region. So this all which surrounds the ascending and descending limb of loop of Henle, we call it the medullary interstitial fluid. It's called medullary interstitial fluid, right? And this will help in maintaining the high osmolarity. So here the meaning of okay, so the meaning of osmolarity is the concentration. So osmolarity is a measure of concentration of solutes in the fluid. So we are talking about especially the concentration of, okay, solutes in that solution, that is in the medullary interstitial fluid, and there will be lot of solutes present, okay, in the medullary interstitial fluid. You can define this osmolarity as the number of moles of solute, the number of moles of solute per liter of solution. So that's what the osmolarity. And the medullary interstitial fluid will have lot many, okay, solute molecules, and because of that medullary interstitial fluid will have high osmolarity. So it has high osmolarity. Okay, then the descending limb of loop of Henle is permeable to water. So here I'll just mention descending limb of loop of Henle. So this is permeable to water. So this is the descending limb of loop of Henle, and it is permeable to water but almost impermeable to electrolytes. So impermeable to electrolytes. Okay, so electrolytes will not be, okay, able to move in and out of this Henle's loop, okay, whereas the water can, right, move across the wall of this Henle's loop, right? And this process actually, so which is permeable to water and impermeable to electrolytes, so this arrangement actually helps in concentration of urine as it moves down. We know the filtrate moves in this direction. Okay, so the arrow marks indicate the movement of the filtrate in the descending and ascending Henle, loop of Henle. And this mechanism, that is descending limb of loop of Henle, which is permeable to water and then impermeable to electrolytes. So this process actually helps in, okay, concentrating the urine, as it flows down. So, right, so as the urine comes down to this, okay, right, the loop, the concentration of the urine increases. Then coming to the ascending limb. So the ascending limb will be, okay, opposite to limb of loop of Henle, and this is opposite to the descending limb of loop of Henle. So here in this case I said permeable to water and impermeable to electrolytes, whereas the descending limb will be, right, permeable to water. I'm sorry, will be impermeable to water. So it will be impermeable to water and it will be permeable to the electrolytes and permeable to electrolytes. It means that the electrolytes can, okay, move into the loop of Henle out of it. So here, right, this that is as I said, it is permeable to electrolytes, and these electrolytes will move either passively or actively. So the electrolytes move either passively or actively, which means in the case passively, which means it doesn't use energy for the movement of the electrolytes. So passively, okay, uh, doesn't use energy for the movement of those electrolytes, whereas actively use energy. Okay, later we'll talk in detail about the counter current multiplier system. So there we will see in detail that whether the water, okay, from the loop comes out or enters into it, okay, or the electrolytes move in or come out. So what actually and how it all takes place, I will detailedly discuss in counter current multiplier system. Okay, so at present I'm just talking about the functions of okay, different tubules of the nephron. So here the descending limb is permeable to water and impermeable to electrolytes, and the ascending limb is impermeable to water but permeable to electrolytes. So now because of this, by the time the urine comes to this loop, it will be highly concentrated, okay, which means it will have more number of solutes. And as it moves up, okay, in the ascending limb, okay, what happens is it gets diluted, okay, due to passage of electrolytes from the limb to into the medullary interstitial fluid. Okay, so this mechanism helps in diluting urine. So the mechanism helps in diluting urine as the urine moves upwards, and this is due to the passage of the electrolytes. So when the electrolytes come out of the limb, okay, it will be left with the water, and water cannot come out because it is impermeable to, okay, water. That's all about the functions of the Henle's loop that have a descending limb and ascending limb. The next one we'll see about the DCT. So distal convoluted tubule. So coming to this distal convoluted tubule, it is lined by cuboidal epithelium. So it is lined by cuboidal epithelium. Unlike in case of the PCT, the cuboidal epithelium have microvilli, but in this case the cuboidal epithelium will not have microvilli. So it is lined by cuboidal epithelium without, okay, microvilli. Now let's see the functions of the distal convoluted tubule. So in short we call it DCT. Now, okay, depending on the body requirement, okay, in the DCT, sodium and water is okay, reabsorbed. So depending on the requirement, sodium and water, okay, are reabsorbed in this segment. So that is distal convoluted tubule. And then the DCT also has a capability to reabsorb the bicarbonates. Okay, it is also capable of absorbing bicarbonates. It absorbs bicarbonates. Then in the distal convoluted tubule, there will be selective secretion takes place. So selective secretion, the name itself tells that, okay, selectively some substances will be secreted, means released out. So that will be, okay, like these hydrogen ions, and then potassium ions, then ammonia, okay, will be secreted, and this actually helps in maintaining. So the selective secretion helps in maintaining the pH, okay, and also it will help in maintaining sodium potassium balance in the body, and also sodium potassium balance in the body. Okay, so here one thing we have to keep in mind, so during the filtration, not just the waste materials will be eliminated. So many of us think that during the glomerular filtration only that urea or uric acid or some other, okay, excretory products will only get filtered, but it is not the fact. Okay, the fact is many useful substances will be also filtered from the blood. Now once these are removed from the blood, depending on the requirement of the body, okay, some of these substances the body will try to reabsorb them back. So how that reabsorption and everything we will see in the coming sessions. Okay, right. And this will actually help in maintaining the electrolytic balance in the body fluids, and also the water balance in the body. If already there is enough water in the body, then there will be, okay, release of water to outside, so in the form of urine, so the person will have a diluted urine. Okay, and if someone have taken some excess, okay, food that may have excess sodium, potassium, okay, or results in the formation of ammonia, and so then there will be a release, okay, more quantities of hydrogen, potassium and ammonia will be released out. And if it is less, then it will try to reabsorb back these substances, and whatever, it will try to maintain a homeostasis of the body. So that is the function of the kidneys. Okay, so selective secretion, which simply means selectively it will release the substances into the filtrate, like these hydrogen, potassium, okay, ammonia, and that will help in maintaining the pH and also sodium potassium balance in the right body. So that's about, okay, some functions of DCT. Then let's look at the functions of the collecting duct. Functions of collecting duct. So let's see the functions of the collecting duct. So from the collecting duct, large amounts of water can be absorbed. So large quantities of water is reabsorbed from this segment, that is, okay, collecting duct, and this will help in, okay, producing more concentrated urine. So this results in producing concentrated urine. Okay, then this segment will also allow the passage of small amounts of urea into the medullary interstitial fluid. Okay, this actually help in maintaining the osmolarity. Okay, so another function is, so amounts small amount of right, urea is passed into medullary interstitial fluid. Passed into medullary interstitial fluid. I told you that in the medullary region, so these tubules are all, okay, surrounded by the fluid, and that we call it medullary interstitial fluid. And as small amounts of urea is passed into the medullary interstitial fluid, it will actually help in maintaining, okay, the osmolarity in the medullary interstitial fluid. So this process actually helps in maintaining osmolarity in interstitial fluid. So that is medullary interstitial fluid. Then this segment, the collecting duct, will also play a role in maintaining the pH. So it helps in maintaining pH. It helps in maintaining pH and the ionic balance of body. Okay, and this it will do it by selective secretion of hydrogen and potassium ions. Helps in maintaining pH and ionic balance, and it is by, okay, selective secretion. So already I told you what is the meaning of this selective secretion of, right, potassium and hydrogen ions. So this is how the collecting duct will actually help in maintaining, okay, the osmolarity in the interstitial fluid, help in producing the concentrated urine, and also helps in selective secretion of these potassium and hydrogen ions. Now let's see the diagram of the nephron and see at what parts the absorption takes place and selective secretion takes place and so on. Right, so here I have drawn the diagram of the nephron, and in the earlier session I already told you that, okay, the PCT and the DCT and some portion of the collecting duct will be present in the cortex region, and this all from here, so below the cortex, okay, so all deep, this is the cortex, I mean, sorry, this is the medullary region, where you can differentiate into outer and inner medullary regions, and you can see this is the collecting, okay, duct that finally opens right at the tip of the renal papillae. Okay, and this portion is the PCT. So I'll just mention here PCT. This is the proximal convoluted tubule, and this portion is DCT, the distal convoluted tubule, and this is the Henle's loop. And here this Henle's loop, they have mentioned, okay, this as the thin segment of a loop of Henle, and above that we have is, okay, thick segment of ascending limb. So thick segment of ascending limb and thin segment of ascending limb. I told you that the thick and thin segments we will see only in the case of juxtamedullary nephrons, where the loop of Henle is very long, and in case of cortical nephrons the Henle's loop is very short, and in that case the cortical nephrons will have only the thin segment. So by showing the thick and thin segments, we indirectly come to know this is the diagram that I am talking about, is a juxtamedullary nephron. Okay, so right, and this is the descending limb of loop of Henle. So this is the descending limb, okay, and this is the ascending limb of loop of Henle. So now let us see at different regions of this nephron. So what parts, I mean, what substances are absorbed and what substances, okay, are excreted. So here the, you know, the bicarbonates will be absorbed from the PCT. So this arrow indicates the absorption, okay, of the bicarbonates from the PCT region, and from the PCT region it also absorbs sodium chloride. So when I put this arrow mark in this way, it is absorbed from the PCT, okay, that is proximal convoluted tubule, into the blood circulation. Because of course first into the interstitial fluid, from there it will be into the uh, right into the blood circulation. We know this PCT, the Henle's loop, and the DCT are all covered by the blood capillaries, that what we call peritubular network, which is formed by the efferent arteriole. So efferent arteriole forms a network, okay, of blood vessels on PCT, DCT, and even on Henle's loop. Okay, so because of that, what happens from this, whatever the substances reabsorbed, so they first enter into the interstitial fluid, from there they enter into the right blood capillaries. So here bicarbonate is reabsorbed, and sodium chloride is also reabsorbed from here, and also the water is also reabsorbed from the PCT, and also the nutrients also absorbed from the PCT, and also the potassium ions. Now what passes into the PCT? Okay, so hydrogen ions from the interstitial fluid, it enters into the right PCT. Then also ammonia from the interstitial fluid, it enters into the PCT. So this is, okay, release of substances into the PCT, and these are all the substances that came out from the PCT, right? And uh, coming to the descending limb of loop of Henle, so we know that it is permeable to water, so hence the water enters into the interstitial fluid. So you have to keep in mind, so all these tubules are surrounded by fluid, and we call it interstitial fluid. So the fluids what you see around this Henle's loop, as the Henle's loop is present in the medullary region, we call it medullary interstitial fluid. Okay, and okay, the ascending limb is permeable to, okay, electrolytes. So you can see the movement of sodium chloride into the interstitial fluid. So if it is necessary for the body, they will be absorbed into the peritubular network, so which is formed by the efferent arteriole. Okay, and then it returns back to the blood circulation. Then, okay, coming to the DCT, right? Here the potassium will be released into the DCT, and then also the hydrogen ions will be released into the DCT, and from the DCT, right, sodium chloride will be reabsorbed. Okay, and also it reabsorbs bicarbonate ions, and also it reabsorbs water. So sodium chloride, bicarbonates, and water are reabsorbed from the, okay, the DCT region. Clear? And here I'll show you direction of a flow of okay, filtrate. So the filtrate, okay, from this, okay, it moves in this direction. Okay, so this indicates the flow of, okay, urine or the filtrate. So here these are all the one who will be connected to other DCTs. So here you will see other DCTs connecting, so which we have removed and showing only the collecting duct. Okay, so here in the DCT, the potassium and hydrogen ions will be released into the DCT, and from the DCT the sodium chloride bicarbonates and water will be reabsorbed. One important point here, so in each kidney, so there are about 250, okay, right, large collecting ducts, and each collecting duct will, okay, take up, the DCTs from around 4,000 nephrons. So each collecting duct, okay, uh, right, collects urine from, okay, 4,000 nephrons, right? So here, so it is connected to, so all this is from another nephron. Okay, this is from another nephron. So like this you will find around 4,000 nephrons. So this collecting duct will take up, okay, the urine from 4,000 nephrons. Okay, right. Then coming to this collecting duct, the collecting duct also, right, it releases, okay, I mean from the collecting duct, okay, urea will be released into the interstitial fluid. Some of this urea will remain in the medullary interstitial fluid to maintain the osmolarity. Okay, so for that, in countercurrent mechanism we will see what is the importance of maintaining high osmolarity in the medullary interstitial fluid. Okay, then from here, okay, the water is also reabsorbed. So just now we have seen the functions of it. Now some portion of that urea which is released, okay, by the collecting duct and entered into the interstitial fluid. So see, I told you, I think now by many times, that this all, okay, tubules are completely surrounded by interstitial fluid. So it is actually released into the interstitial fluid. From there, the some portion of the urea, okay, will be passed into the thin segment of ascending limb, which will be for the process of excretion, means release it to outside. So this is actually the reabsorption and secretion of different substances. So the hydrogen ions and ammonia is secreted into the PCT, and there is a HCO, I mean that is bicarbonate, sodium chloride and water, and potassium ions and nutrients, these are reabsorbed. So this arrow indicates a secretion and this indicates, okay, reabsorption. Okay, right. So that's about reabsorption and secretion of some major substances at different locations of the nephron. Next session we will see in detail about the urine formation.